Today we will discuss about lock and key theory of enzyme. What is enzymes? Enzymes are protein molecule. There are made up of amino acid. How we will define that enzymes? Enzymes are biocatalyst. It speed up rate of reaction in our body. This is enzyme. Enzymes are biocatalyst. That means speed up biological reaction is the cell but remain unchanged at end of the reaction. This is substrate. Substrate going to be break down by the enzyme. Our cells is actually very busy place and most of the work does if more molecules around or change molecule other kind of molecule by either making smaller molecule bigger or bigger molecule smaller, and all this reaction required something called enzyme as a traffic police to make things faster, and there are not destroyed or change of the reaction. The action of enzyme can be explained by lock and key theory or hypothesis. Enzyme act as a lock because each enzyme has a region very specific shape is known as active site. This one is enzyme, this one is active site of enzyme. This is substrate. It is completely complementary to active site of enzyme. This one is active site of enzyme. This one is the complementary to active site of enzyme. So it fit into the active site like a key to fit into the lock. So substrate act as a key. This one enzyme, this one substrate. Here it will fit to the active site of the enzyme. So lock is enzyme, key is the substrate. This one is lock is enzyme, key is the substrate. The only size key fix into the key hook of the lock. It means the enzyme specifically react with a very similar component. Okay, enzyme will react with a very similar component only. Once it attached to the enzyme, the whole thing has become enzyme substrate complex. Enzyme substrate, here it will attached means the whole thing name is called as enzyme substrate complex. So the reaction will happen, the bond in the substrate molecules are broken here. The reaction will happen, the bond of the substrate molecule here will broken. Here broken. Once the bond are broken, the product will be formed. Broken of this bond, the product will be formed. The product no longer fit into the active site and they escaping into the surrounding medium, leading the active site free to receive other substrate molecule again. So as you see here, the structure of enzyme molecule still remain unchanged and it can bind other substrate molecule for other reaction. Third steps, enzyme breakdown substrate into product. Okay, here product. So enzyme breaks substrate into the product. Enzyme substrate complex, after that it produced product. This is about that lock and key theory of enzyme. Thanks for watching.